In class, we discussed cell cycle control and how we regulate the cell cycle. One of the main things we talked about were CDKs and cyclins. So this video is to finish up the notes on internal signals and also to model it for you so that you can see what's actually happening in the cell. So as we discussed in this graph, the CDK levels stay constant throughout the entire process, but cyclin levels change. So the question that we asked in class today is, what do CDK and cyclin complexes do? And I gave 3 specific examples. The first specific example I gave was all about the G1 complex. The G1 complex is a combination of a G1 cyclin and a CDK. And what the G1 complex does is it phosphorylates DNA so that it can replicate, which pushes the cell from G1 to S. When it phosphorylates the DNA, it energizes it so that replication can occur. And that's why the cell cycle, when the G1 complex is present, the cell can go from G1 to S. This only happens if G1 happens correctly. What do I mean by that? Let's take a look at this cell I set up. In this cell, we can see that the cell has started to grow. Okay, so right now we are in G1. The cell has started to grow. Okay, if the cell grows, we know that inside a cell, the CDK levels are constant the entire time. So CDK levels aren't going to change. But you get a cyclin when the cell does something right. So in G1, the cell grows. Congratulations, cell. You get a cyclin, a G1 cyclin in this case. Let's say the organelles replicate. We make more mitochondria. Congratulations, you get a cyclin. Let's say we make more endoplasmic reticulum. Congratulations, you get a cyclin. So when the cell does things it's supposed to do in G1, it also naturally makes cyclins. These cyclins at this point are called G1 cyclins. They're going to attach to the CDKs. Now the CDKs are active. So the CDKs were always present in the cell, but they were inactive. So as we make this G1 cyclin, they become activated. What these CDK cyclin complexes are going to do in G1 is they're going to go to the nucleus, to the chromatin in the nucleus, and they're going to phosphorylate it. Boom, phosphorylated. Boom, phosphorylate the chromatin. Boom, they're going to phosphorylate the chromatin. After they do that, the cyclins are going to break down and be degraded, but the CDKs are still going to be present. Once this DNA is energized enough, okay, it's going to be able to start DNA replication. So having these CDK cyclin complexes phosphorylate the DNA is going to push this into the next phase, from G1 into S when the DNA is replicated. So if we look at our big sheet, we can see during G1, if the cell is growing appropriately, that means the G1 cyclin is going to be being made. And when we have enough G1 cyclin binding with the CDK, so this is G1 cyclin, when we have enough of the G1 cyclin and CDK complex, okay, we're going to phosphorylate the DNA, and then the cyclin is going to be broken down, but the CDK is going to remain constant. But that's okay because we've got over that hump. We were able to push from G1, which is growth, to S, which is DNA synthesis. Okay, so that's our first checkpoint passed. The second checkpoint we're going to be talking about is what happens. So that's our G1 complex. The second checkpoint we're going to be talking about is the G2 checkpoint, and this is going to use our G2 complex. The G2 complex, just like the G1 complex, is going to be made up of CDK, But now we're going to use the G2 cyclin. The G2 complex is going to phosphorylate the nucleus, causing it to break down. And this is going to push the cell from G2 into mitosis. And let's look at my cell so I can show you what this looks like. So now we've finished G1, the DNA is replicated, we've finished synthesis. Now we're in G2. The DNA is replicated, okay, and we want to move into mitosis. Well, our G2 cyclin will have been building up. So when the DNA replicates during synthesis, we make a G2 cyclin. As the DNA continues to replicate, we continue to make G2 cyclins. As we proofread the DNA, we continue to make G2 cyclins. Okay, so we've made all of this G2 cyclin. Once again, the CDK was always present, but the G2 cyclin started to build up and join to its CDK, because there's different kinds of CDKs. So now we have a G2 cyclin plus a CDK combined. That is a complex of cyclin and CDK, and that is going to phosphorylate the nucleus. And these phosphate groups that are added to the nucleus are added from the CDK cyclin complexes. As this complex phosphorylates the nucleus, the CDK and cyclin split, this G2 cyclin is broken down, but of course the CDK remains constant. But its work has been done. This nucleus is phosphorylated, and these phosphate groups kind of act like nuclear bombs. They break down the nucleus so that the DNA can leave the nucleus and begin to form chromosomes so that we can move from G2 into mitosis. Now let's look at the third checkpoint, the most important one in terms of examples, which is the M cyclin plus CDK complex called MPF. Luckily, we wrote most of this information down in class, so if you missed it, just go ahead and write it right here. The third checkpoint we're looking at is right here. This is number 3. This is called the M checkpoint, or this is called the M complex. The M complex is made up of M cyclin plus CDK. That complex is known as MPF. And like we discussed earlier, as the cell correctly goes through synthesis and alignment, M cyclin will build up, okay, and will attach to a CDK to form MPF. The MPF is going to phosphorylate the spindle fibers, causing them to shorten, causing the cell cycle to pass from metaphase to anaphase. So let's look at our cell picture again so we have a better idea of what this looks like. So here we are in mitosis. We're kind of in late prophase. You can see we have 1, 2, 3, 4 duplicated chromosomes. Okay, there's no nuclear membrane. These chromosomes are just in the cytoplasm of the cell. Our CDK is constant. But as our DNA was replicated in synthesis, we make M cyclin. As our chromosomes formed, we'll make M cyclin. As the chromosomes begin lining up in the middle of the cell for metaphase, we'll make M cyclin. As the spindle fibers attach to the chromosomes, we'll make M cyclin, and eventually we'll reach this tipping point, right? Because for all these complexes, you need a tipping point where we have enough of this complex here. So what will happen is this new complex, which has a special name, MPF, will come to the spindle fiber and will phosphorylate the spindle fiber. Once that happens, the CDK will stay and the M cyclin will be broken down. Now another MPF will phosphorylate a spindle fiber. Oopsies. Once that happens, the cyclin's broken down and the CDK stays. Again, another MPF will phosphorylate a spindle fiber. Okay. As it phosphorylates the spindle fiber, the cyclin's broken down, the CDK stays. So now all these spindle fibers have been phosphorylated. Right now, we are in alignment. We're in metaphase. Once the spindle fibers are phosphorylated, they will begin to shorten. As they shorten, they will pull apart the two sides of the duplicated chromosome. Okay, and that's how we move from alignment, which is metaphase, through that checkpoint to anaphase. And eventually these chromosomes will get together in a nucleus for each cell, and eventually that's the end of mitosis, and then we'll get into cytokinesis where the cytoplasm splits. And of course, there are more checkpoints along that way as well. But you can see the CDK concentration stays constant. As the cell does things right, it makes cyclin, which helps push it to the next phase of the cell cycle. Now, what happens if these CDK cyclin complexes are ignored? Or what happens if the cell does something incorrectly? Well, let's say that the cell does not replicate its DNA correctly. Well, then that cyclin won't build up, and the cell either will have to fix its DNA to move to the next phase of the cell cycle, or it will undergo apoptosis or programmed cell death. Okay? Now, cancer cells ignore these signals, and that's why they divide out of control. So let's look at external signals and how those help control cell division, and also let's look at the p53 gene, which is an excellent example of a protein, the p53 protein, that's a master regulator for this process. So CDKs and cyclins are internal signals inside the cell. But how does the cell get a message from outside? Those are external signals. Okay, and we're going to look at 3 examples: nutrients and growth factors, density-dependent inhibition, and anchorage-dependent inhibition. Okay, nutrients and growth factors, these are ligands or chemicals. Okay, and these ligands or chemicals are released by damaged cells a lot. And they travel to healthy cells to tell them to divide. Okay? So let's say you have like a cut or something, like you cut yourself, and some of the cells are damaged. They're going to send signals to nearby healthy cells saying you should divide and repair this gap. We also do this for growth, right? Like sometimes cells send signals if we need to grow bigger. So like for growth, a good example is HGF. Okay, HGF is a signal we use for growth. And a good example for repair is PDGF. We use this for repair. So this is telling the cell, please divide. Okay? Now, density-dependent inhibition and anchorage-dependent inhibition, these aren't ligands, but they use chemical messengers to make these things happen. Okay? Density-dependent inhibition is going to use chemicals to prevent cell division when cells are too crowded. So if cells are too crowded, they'll send signals to other cells nearby to say, don't divide, there's too many of us, like let's just chill out for a second. Okay? Anchorage-dependent inhibition is kind of the same thing. Basically, it's saying cells must be attached to other cells to divide. So basically, free-floating cells should not divide. Okay? And so it's because cells that need to get messages from other cells to divide, cells get messages from other cells to not divide as well. Now, basically, for both of these, cancer cells do not follow these rules. Cancer cells do not follow these rules. They divide when they're too crowded, and they also divide when they're free floating, which is called metastasis. Okay, so that's kind of, cancer cells ignore these signals. Our bodies can actually combat cancer because we have some really cool regulatory genes and regulatory proteins. Okay, the P53 gene produces a protein that repairs damaged DNA or leads to apoptosis, which is programmed cell death when a cell can't repair itself. So this protein works a lot during G2. Okay, so we do have these proteins that help. That's it for this video. I hope you enjoyed it. Please watch the next video on meiosis as well.